Hi, I'm Bree.
And I'm Justin, and we're studying the GABA-like immunoreactivity on the central nervous system of the blue crab here at Valdosta State University.
Gamma-aminobutyric acid, or GABA, is a major inhibitory neurotransmitter in the central nervous system of many organisms. Here, we're examining the distribution of GABA-like immunoreactivity in the central nervous system of the blue crab, Callinictes sapidus. The central nervous system of crustaceans includes structures like the brain, thoracic ganglion, pericardial organs, and cardiac ganglion. Shown here is a simple schematic of the central nervous system of a blue crab. The crabs used are bought locally and maintained at Valdosta State University in an aquarium. Live crabs are placed under ice, which acts as a natural anesthetic. We begin the dissection by removing the dorsal carapace to expose the crab's internal organs. We then remove the heart, which contains the cardiac ganglion. Next, we remove the stomach and intestinal tracts to extract the thoracic ganglion. The brain is the next organ to be dissected, followed by the two pericardial organs. After the preps have been removed from the crab, they are fixed in paraformaldehyde for 4 hours. They are then washed with phosphate triton azide buffer, which is also known as PTA. Next, the preps are incubated with a primary antibody which binds to GABA for 3 days. They are washed again with PTA and then incubated in a secondary antibody that is conjugated to a fluorescent marker for 1 day. The secondary antibody binds to the primary antibody, which allows us to trace GABA throughout the central nervous system under a fluorescence microscope. We are performing these immunohistochemical experiments to try and find the location of cells in the nervous system which may contain GABA that project to the cardiac ganglion. Here we see the brain, thoracic ganglion, pericardial organ, and cardiac ganglion after being dissected. The preps have been pinned to a petri dish and are ready to begin the series of washes.
GABA-like immunoreactivity was distributed widely throughout the central nervous system. There are many cells found in different locations throughout the thoracic ganglion, and numerous varicosities and fibers were observed within the pericardial organs. The distribution of GABA-like immunoreactivity in the pericardial organs and the cardiac ganglia suggests that GABA could have both a hormonal and neuronal role in the cardiac system of the blue crab. The presence of varicosities in the pericardial organs suggests that GABA could be released here in a hormonal fashion. The fibers running into the cardiac ganglion suggest that GABA could be the neurotransmitter of one of the cardioregulatory fibers. Future research could include further experiments to better optimize GABA-like immunoreactivity on the brain and thoracic ganglion.
Physiological experiments could also be performed on the heart and cardiac ganglia to confirm the inhibitory role of GABA in the cardiac system.
Thank you for watching.